The first step in creating a genetically modified plant is to identify that altering the plant's genes is the best way to improve it, and then identifying the gene or trait you want the plant to have. For example, scientists and agricultural workers in Hawaii were troubled by the papaya ring spot virus in their papaya crops. After failure trying to control the virus with insecticides to eliminate the insects spreading the virus, and delivering a mild strain of the virus to try and induce natural resistance, the scientists decided the best thing was to try transgenics, and needed to find a gene for PRSV resistance. The next step is finding, no, not Nemo and Dory, the next step is finding a suitable organism that already has the desired genes. In the case of PRSV resistance, it was found that inserting genetic material of the virus into the fruit's DNA created resistance. There are two common methods for transformation of genes from one donor organism to another. The first we will look at is biolistics. This involves using a gene gun. This, unfortunately, I know, is not a gene gun. But ta-da! This is! The gene gun uses high-speed particle bombardment to shoot metal, usually gold, ooh, shiny, coated in DNA into the plant cells in a petri dish. The cells that took up the desired genes into their chromosomes now multiply to form a callus, then undergo root generation followed by shoot generation until finally a GM plant with the new desired trait is grown. The other common method is Agrobacterium transformation. This uses a natural plant pathogen called Agrobacterium tumefaciens that contains a TI plasmid. The TI plasmid is cut using restriction enzymes and the natural tumour-inducing genes are removed. The desired gene and a biological marker, often an antibiotic resistance gene, are inserted and the recombinant plasmid is joined together by DNA ligase. The recombinant TI plasmid is then introduced into plant cells in culture, which are grown on an agar plate that contains ampicillin, an antibiotic. The cells that successfully took up the desired genes will have the genes for antibiotic resistance and survive, hooray! And the unwanted non-recombinant cells will die, and a new plant with the desired genes is regenerated.